Hello everyone, my name is Li-Chun Chen. I am the Chief Scientific Officer for CYTO Bio. Today I'm in Denver attending CYTO 2025. We are presenting poster today. One of the poster titled Unsupervised Clustering Reveal Cell Types in 40 Specimen, GVHD PBNC Cytometry Dataset. This is a work that we collaborate with University of Minnesota, with Dr. Sherman Holden's group. And this 40 samples come from 20 patients that experience acute graft versus host disease and undergo trophing and EGF treatment. The cohort is evenly balanced with half of the patient in the responder group, half in the non-responder group, and we also have baseline and on-treatment time points. Just to give everybody a little bit of background, gating has been the major tool to analyze cytometry data, but gating can start to get really labor intensive when your cytometry panel starts to have higher and higher number of markers. For example, to gate a cytometry dataset with 44 marker panel, in theory, they will yield almost billions of potential combinations, and that require hundreds of hours to truly fully gate the data set. To overcome that, computational approach, including unsupervised clustering analysis, have been used by many groups as an alternative to analyze high-dimensional cytometry data set. So here we are going to present one of the clusters that we identify from using this method. We generated the FCS file using mass cytometry assays using this 44 marker panel. We do some manual gating in parallel just to compare the performance of our unsupervised clustering. And for the computational method, we use fast phenograph to do the clustering, and followed by an in-house built tool to name each cluster. So I'm gonna go more detail in terms of this cluster naming tool in a bit. And after that, we also manually QC the cluster by looking at the heat map and then identify protein marker that was distinctively expressed in each cluster. And with this method, we are able to identify 48 clusters from this data set. By using the cluster naming algorithm, we are able to match them using a similarity matrix to name each cluster with the best knowledge that we have using gating. When we match the 48 cluster with our 36 gated population, some of the population may be overlapping, partially overlapped. Then we evaluate the expression pattern. So here we're gonna have you focus on this cluster. We call them cluster 26. So cluster 26 is a small cluster that was within the CD4 T cell bigger island. And when we look at this cluster 26, we found that it shares some similarity with the CD4 TEMRA cells, with their expression of CD27 and CD45RA. So by looking at CD27, it is the negative population. But when we look at the CD45RA, it has intermediate expression compared to the gated TEMRA cells, which is on the other side of this island. So we then focus on a marker that highly expressed within this cluster. And we found that this cluster is mainly defined by the expression of KRG1 and Tibet. So we then named this cluster KRG1 positive, Tibet positive CD4 T cells. And we then further look at how these clusters express other functional marker and whether they show some significant differences between responder, non-responder, or time points. And we found that CD25 expression within this cluster are significantly different at the baseline between responder and non-responder. So to conclude, we are able to use unsupervised clustering method to identify a new group of cells that we probably will not identify by traditional gating. And if you are interested in discussing what is the implication of this cluster, please come to my poster on number 263. And you can also view this data set on f.teco.bio/projects UMN002. Thank you for your time.